A few years ago, with my colleague Emmanuel Charpentier, I invented a new technology for editing genomes. It's called CRISPR-Cas9. The CRISPR technology allows scientists to make changes to the DNA in cells that could allow us to cure genetic disease.
In 2020, the Royal Swedish Academy of Sciences awarded the Nobel Prize in Chemistry to Emmanuel Charpentier and Jennifer Doudna for their work on CRISPR, a method of genome editing. CRISPR technology is adapted from a system naturally present in bacteria and archaea, which Charpentier and Doudna were both studying independently prior to their joint award-winning discovery. Born in 1968 in France, Emmanuel Charpentier trained in the studies of biology, microbiology, biochemistry, and genetics at multiple well-known institutions, including the Institut Pasteur and St. Jude Children's Research Hospital in Memphis. Charpentier is director at the Max Planck Institute for Infection Biology in Berlin, honorary professor at Humboldt University, and visiting professor at the Laboratory for Molecular Infection Medicine in Sweden. Jennifer Doudna spent much of her youth in Hilo, Hawaii. After earning a degree in chemistry from Pomona College in California, she went to Harvard University. There, she worked in the laboratory of an English-born American biochemist and geneticist Jack W. Szostak, who won the 2009 Nobel Prize for Physiology or Medicine. In 1989, she completed her PhD in biochemistry. Eventually, in 2002, she moved to the University of California, Berkeley, where she served as professor of biochemistry and molecular biology. While studying Streptococcus pyogenes, Charpentier discovered that a molecule called TrkRRNA was part of bacteria's ancient immune system, CRISPR/Cas, that cleaves viral DNA. In bacteria and archaea, CRISPR and its associated proteins constitute an adaptive RNA-mediated defense system. Essentially, bacteria and archaea acquire resistance to viruses and plasmids by integrating short fragments of foreign DNA into clustered, regularly interspaced, short palindromic repeats, or CRISPRs. These repetitive loci maintain a genetic record of all prior encounters with foreign transgressors. CRISPRs are transcribed, and the long primary transcript is processed into a library of short CRISPR-derived RNAs, or cRRNAs, that contain a unique sequence. Charpentier's study uncovered a novel pathway of CRISPR activation wherein a trans-encoded small RNA, the host endoribonuclease 3, and the CRISPR-associated CSN1 protein are responsible for the production of the active cRRNAs. Meanwhile, Doudna was deciphering the structure and biologic function of RNA enzymes and recognized the same RNA-based surveillance system in bacteria. In E. coli, cRRNAs are incorporated into a multi-subunit surveillance complex called cascade, or CRISPR-associated complex for antiviral defense, which is required for protection against bacteriophages. Using cryo-electron microscopy to determine the sub-nanometer structures of cascade before and after binding to a target sequence, Doudna's research found a conformational rearrangement that could serve as a signal that recruits a transacting nuclease, Cas3, for destruction of invading nucleic acid sequences. After meeting at a conference, Charpentier and Doudna had a eureka moment, realizing that jointly they could harness the RNA-based complex to develop a novel gene editing system with the power of precision. They found that in a subset of these systems, the mature cRRNA base paired to transactivating TrkRRNA forms a 2-RNA structure that directs the CRISPR-associated protein Cas9 to introduce double-stranded breaks in target DNA. At sites complementary to the cRRNA guide sequence, the Cas9 HNH nuclease domain cleaves this complementary strand, while the Cas9 RUVC-like domain cleaves the non-complementary strand. Ultimately, the study revealed a family of endonucleases that use dual RNAs for site-specific DNA cleavage and highlights the potential to exploit this system for RNA-programmable genome editing. Charpentier and Doudna were the first to define an in vitro CRISPR-Cas9 system that not only could cut viral DNA, but could also be programmed with an engineered guide RNA to cut any DNA at a sequence-specific site. According to Doudna, following the publication of the initial CRISPR-Cas9 study, at least 6 papers describing different uses of CRISPR-Cas9 for genome engineering had been submitted for publication. By the end of 2014, scientists had, among other things, used CRISPR-Cas9 to enhance pest resistance in wheat, reproduce the carcinogenic effects of specific chromosome translocations in mouse lungs, and correct a mutation in adult mice that in humans causes the hereditary disease tyrosinemia. From the rapid success, however, came an ethical debate surrounding the potential uses of the technology. Following numerous publications, including a 2014 study in which researchers described how they had used CRISPR-Cas9 to make precise changes to the genomes of cynomolgus monkey embryos, which are so genetically close to humans that they are commonly used to model human genetic disease, Doudna decided to enter the public discussion regarding the ethics of their own creation. After discussing the issues surrounding the modification of the human germline, they published a perspective article in Science that urged the global scientific community to refrain from using any genome editing tools to modify human embryos for clinical applications. They also recommended that public meetings be convened to educate non-scientists and to enable further discussion about how research and applications of genome engineering might be pursued responsibly. That being said, today, CRISPR is still the preferred gene editing tool for thousands of researchers. In October 2016, a Chinese team announced they had introduced modified T cells into a patient suffering from an aggressive form of lung cancer. CRISPR-Cas9 had been used to modify PD-1, a cell surface receptor usually expressed on T cells that dampens the immune response. Thus, if present on cancer cells, it prevents them from being attacked and cleared by immune cells. Knocking out this gene would allow a full immune response against the tumor cells to be mounted. The technology holds similarly vital potential in streamlining agricultural and food production. For example, a 2018 study targeted cacao trees due to their susceptibility to fungal diseases prevalent in West Africa. Previous work in cacao identified a gene, known as TCNPR3, that suppresses the plant's disease response. The researchers hypothesized that using CRISPR-Cas9 to knock out this gene would result in enhanced disease resistance. To test their hypothesis, they used Agrobacterium, a plant pathogen modified to remove its ability to cause disease, to introduce CRISPR-Cas9 components into detached cacao leaves. Subsequent analysis of treated tissues found deletions in 27% of TCNPR3 copies. When infected with Phytophthora tropicalis, a naturally occurring pathogen of cacao and other plants, the treated leaves showed greater resistance to the disease. The results suggested that the mutation of only a fraction of the copies of the targeted gene may be sufficient to trigger downstream processes, resulting in systemic disease resistance in the plant. With CRISPR-Cas9's prevalence and established vitality in altering the future of biotechnology across numerous disciplines, some researchers have gone so far as to call the 21st century the age of CRISPR. Ultimately, Charpentier and Doudna's revolutionary gene editing technology encapsulates the mantra that with great power comes great responsibility.